Today we're looking at Chapter 12, Part B, so we're focusing on plant structure and function. Let's talk about leaves. Leaves are the location for photosynthesis, and they capture light energy and convert it into chemical energy in the form of carbohydrate. A input would be carbon dioxide, and an output would be oxygen. The photosynthesis turns that carbon dioxide plus water into sugar and oxygen. We can also look at the venation of a leaf. It tells us something about the taxonomy of the plant. We see parallel venation in monocots and branched or netted venation in dicots. Continuing to look at the macroscopic view of leaves, we see the blade, that's the primary portion of the leaf. The apex or the point is the tip of the blade, where the blade attaches to a stem, that is a stalk called the petiole. And then we can see below the petiole stipules, which are small stalk-like or even leaf-like growths that would protect an early leaf bud. Monocots again have parallel venation. Dicots have a netted branched pattern. And so this base taxonomy is actually going to be indicative of how the leaves look in a plant. Continue to look macroscopic, we can compare parallel to the netted venation. Now, the parallel venation is when a leaf's veins run up and down the leaf parallel to the midrib, with the midrib being the primary vein in a leaf. And if you look closely, you'll see that these veins tend to run all throughout leaves, and they're composed of xylem and phloem, and they just form these different patterns in different leaves. If it's not the parallel venation, it might be branched. There's pinnate, where we have a midrib with veins that branch from that midrib. And if the branches off the midrib have branches, then it's going to be not pinnate venation, but palmate venation. If we look closer at leaves, we can look at the microscopic view. There is a top and bottom layer made up of epidermal cells that are clear. Sometimes the epidermal cells secrete a waxy substance that would be called the cuticle, and it makes leaves shiny, whereas leaves that don't have cuticles usually grow hairs, and that would give them a velvety appearance. Now, on the underside of leaves, in the lower epidermis, is where you would find holes called stomata. They are there for gas exchange with the atmosphere. Each stomata is guarded by two cells, very conveniently called guard cells, and they open and close the stoma. But when these guard cells swell, they bend out, opening it, and when they shrink, they collapse, and that would close the stoma. In order to open the stoma, there is a chemical mechanism. Low carbon dioxide levels and sunlight actually trigger the guard cells to pump an abundance of potassium ions into their vacuoles, causing the inside of the cells to become hypertonic. Water follows the potassium ions, and that's going to cause water to enter the vacuoles of these cells as well. And therefore, they're taking up water by osmosis, and they are swelling up and becoming turgid. And so that makes their inner walls rigid, and they be pulled apart, and this would open the pore or the stoma. So here we can see the schematic of stomata. The opening is the stoma, and surrounding are the guard cells. Something like oxygen and water vapor could leave the stoma. Something like carbon dioxide could also go in and out, but oftentimes goes in order to participate in metabolic pathways like photosynthesis. Here's a better view of the guard cells open and closed. They open the stoma when they're swollen, meaning they've not only taken in potassium ions, but also water into their central vacuoles. And then when those are deflated, we would see the guard cells shrunken. Now, the amount of light a leaf receives is going to be the most important factor in controlling the guard cells. If there's a lot of light, photosynthesis can take place. That means the guard cells need to open the stomata to allow for the cells in the leaf to take in carbon dioxide. That way photosynthesis can take place, and then release oxygen, a byproduct of photosynthesis. But if the light levels are low, the leaf cannot afford to have the stomata open because you don't want water to evaporate via transpiration from the leaf through the stomata. Transpiration is when water evaporation occurs through the stomata, and when light levels are low, plants cannot perform photosynthesis. So, what's the purpose of having the stomata open? We'd rather see them closed, or the plant would rather see them closed to protect the leaf from excess water loss. And so, temperature and the amount of rain could also impact the opening and closing of guard cells. If it gets too hot, guard cells close to prevent water loss. If there is a drought or it is too dry, guard cells will also close to prevent water loss. Here's what stomata actually look like under an electron microscope. Of course, the one on the left is closed and the one on the right is open. Keep in mind that the stomata with its guard cells are only in the lower epidermis. The base structure for a leaf, again, is an upper and lower epidermal layer with inner cells. The inner cells are called mesophyll cells, and the top layer is palisade mesophyll, where we see cells that are tightly packed together with cytoplasmic streaming chloroplasts. And down here at the bottom, this is the spongy mesophyll, and these are loosely arranged cells with plenty of space for carbon dioxide absorption. Here we can see a cross-section of the veins in a leaf. Remember, the veins that run through a leaf were made up of certain types of tissue. We've talked about two of the three. We talked about xylem and phloem. We didn't talk about that which is not vascular tissue. Number 3, collenchyma. These are living cells that are very thick. They are more supportive in function, and they're going to support the veins. Little collenchyma is found near the margin or apex of the leaf because the veins get so small, but near the midrib you would find quite a bit of collenchyma. Again, this is a support structure for the vascular tissue that's the primary components of the veins. We also want to do a quick monocot-dicot review now that we've finished looking at the venation, and one of the last differences we will establish in taxonomy between monocots and dicots. So monocots have one seed leaf, fibrous roots, scattered arrangement of vascular bundles in the stem, parallel veined leaves, and their flowers have multiples of 3. So 3, 6, those would be acceptable. Dicots have 2 seed leaves or 2 cotyledons. They have taproots. They have a prominent central root. They have ringed vascular bundles in their stem, and they have branched net-like veins. And their flowers have petals that are of multiples of 4 or 5. We can also talk about leaf color, so we have to talk about chlorophyll. Most leaves are green because the chlorophyll in their chloroplast gives them their color. The chlorophyll reflects green and absorbs everything else. The more chlorophyll containing cells in an area of a leaf, the darker the area appears. So why does it make sense for more chlorophyll containing cells to be in the palisade or top layer of the mesophyll cells as opposed to the spongy or the bottom mesophyll layer? Well, the reason we have more chlorophyll in the palisade or top layer of mesophyll cells is because that's closer to the top of the leaf. It's closer to the upper epidermal layer. That's the layer that's going to get the most sunlight, so it makes sense for it to contain the most chlorophyll because chlorophyll absorbs sunlight. But there are other pigments that absorb light that aren't chlorophyll. There's carotenoids, anthocyanins, phycobilins. Carotenoids have a yellow or orange hue. Anthocyanins change color with differing pHs. We see blue, purple, and pinks from anthocyanins. And phycobilins give off red or blue. They are all photosynthetic pigments, but chlorophyll is going to be more prominent in leaves. Let's compare evergreen to deciduous plants. An evergreen plant is a plant that generally has needles, but not always, but it never changes color and stays essentially vegetative all winter. It stays green all year round. A deciduous plant loses its leaves in the winter. They do so to conserve water, and at the base of each petiole in a deciduous plant, there's a thin layer of tissue called the abscission layer, and the cells of the abscission layer perform a specialized task. They note when the days start getting shorter, meaning winter is approaching, and that means that the cells in the abscission layer will begin to activate. They'll block xylem and phloem running through the petiole to the leaf, and once they've succeeded in blocking off essentially the nutrition routes, we'd see them begin to die. As the cells in the abscission layer of the petiole die, they crack, and that cracking becomes so severe eventually that the leaf falls off the tree under its own weight. So the xylem and phloem that is blocked by the abscission layer, but if they're blocked, they can no longer produce chlorophyll. So as a result, we also see the green color of leaves fade as this abscission layer is trying to lead to eventually the leaf falling off the tree. But that means we will start to see colors that were hidden by the abundance of chlorophyll. Will reveal whatever pigments were covered by the vibrant green. So here's an up-close view of the abscission layer. Again, it is located between the petiole and the branch. Let's not forget that we mostly rely on vascular bundles to transport water and nutrients, and a plant depends on water and the dissolved nutrients. But the water needs of plants are definitely different depending on what ecosystem they have been designed to occupy. Water is used for various processes, but four are essential: photosynthesis, development of turgor pressure, hydrolysis, and transport. Photosynthesis is when plants use carbon dioxide and water to make their own food. They're going to turn that carbon dioxide and water into usually a carbohydrate like glucose. And for every molecule of glucose, 6 carbon dioxide molecules and 6 water molecules were used. The development of turgor pressure is important to keep plants upright or in the right position. In plants, there's a large central vacuole in their cells, and that fills with water by osmosis. As more and more water fills the vacuole, plant cells become pressurized, and this pressure causes turgor pressure. It keeps the plant stiff. It doesn't cause the cell to life, because remember, plant cells have cell walls, so they don't lyse, they don't separate or bust. But plants can wilt or they can plasmolyze when they're deprived of water and their turgor pressure decreases or their central vacuoles deflate. Now, some plants can demonstrate nastic movement, and this is movement of a plant or a response to a stimulus such as that of the direction that was reprogrammed or pre-programmed and is not dependent on the direction of the stimulus. For example, some plants open their petals during the day and they close them at night. And that's due to changes in turgor pressure that occur near the base of the flower throughout the day. So again, a nastic movement is a movement that occurs in response to a stimulus but has nothing to do with the direction of the stimulus. I mean, this would occur regardless if the stimulus was coming from the left, right, up, down, or all around. That's a nastic movement. And so the opening of petals is a nastic movement for some plants who open their petals at night. Hydrolysis is another process we want to focus on. This is when a cell takes in large molecules. It needs to break them down first into smaller molecules that can be used. And the cell would do this by adding water to larger molecules. So this would be a hydrolysis reaction. The addition of water actually can break down large molecules. It can break bonds, and that's the process called hydrolysis. And of course, water is great for transport because plants must carry all sorts of biosynthesis and they need water to do so. Chemical reactions occur in the presence of water. The transportation is accomplished by dissolving the materials or ions, other substances needed in water, and then allowing that water to carry itself up the xylem using cohesion and adhesion. The motion of water through a plant actually requires the process of transpiration. Let's talk about that water absorption in plants. Well, it's absorbed by a vascular plant through its roots. That's where water absorption begins. Then the plant can absorb only a small amount of water vapor through its leaves if its stomata are open. But most of the water, again, is absorbed from a vascular plant's roots. And most roots are in the soil, so most roots absorb the water that is in the soil. There are pore spaces in the soil that determine how much water and air the soil can hold. So soil is generally made up of organic substances like mold, bacteria, protozoa, worm, yeast, but also inorganic stuff like gravel, sand, silt, and clay. So both the organic and inorganic components mix. And when you have soil that's made up of larger particles more than smaller particles, think gravel and sand as opposed to something more compact. Then you'll have spaces between those substances, and that would allow the soil to absorb lots of water in a short time, but the water tends to pass through the soil quickly because it's acting essentially kind of like a filter system. That means the soil might dry out soon after it gets wet, but initially it can hold a lot of water. And the soil is made up of smaller particles. That means the soil takes a long time to absorb the water, but once it's wet, it holds the moisture for a long time. It doesn't filter through. If water fills up all the pore spaces in the soil, there's no oxygen for the roots to absorb. And if that condition lasts for too long, they will die. So that's why waterlogged plants die, is because they have too much water and oxygen can't get to the roots. The larger the pore spaces, the more oxygen the soil can hold and more carbon dioxide the roots can release. So a good soil is considered a loam, or a mixture of gravel, sand, silt, clay, and organic matter. And that's what would be needed to create the most fertile soil. So a good loam has some pore spaces that are just large enough to contain plenty of oxygen, but also small enough so that the soil can hold moisture for a reasonably long time without filtering it through and drying out. So the perfect loam that scientists say would give you the most fertile soil would consist of a tiny amount of gravel, 40% silt, 40% sand, slightly less than 20% clay, and the difference should be organic matter. That's the ideal pore space ratio for oxygen and water absorption by roots. They'd be able to absorb plenty of water while still being able to absorb oxygen and release carbon dioxide with this type of loam. Let's also look at ways that water transportation in plants is going to be accelerated. There's three phenomena that seem to be in play to make water move against gravity: transpiration, capillary action, and root pressure. The combination of all three essentially exert a pulling force that draws water from the roots to the tips to the tallest trees. Transpiration is the evaporation of water from the leaves of a plant. Transpiration is evaporation that occurs from inside the leaf when the stomata are open. Transpiration relies on capillary action or the use of cohesion and adhesion. Because water is a polar molecule, it is attracted to other polar molecules as well as itself. Remember, cohesion is the attraction of water molecules to other water molecules, and adhesion is the attraction of water to other non-water molecules. The polar nature of water allows for cohesion and adhesion, and that pull up the plant utilizes those properties, which together get called a capillary action. So essentially, when water evaporates from the leaves, there's a deficit of water in the leaves, and that causes a tension or pulling pressure on the water molecules that are below. And that deficit of water must exist for this capillary action as a part of transpiration to take place, because this tension is often referred to as the transpiration pull. And in the transpiration pull, water molecules respond to the tension by moving up to replace the evaporated water molecules. And so, because of adhesion and cohesion, which allow for capillary action, this transportation pull phenomenon can take place. And the water molecules can move up to stay near the others that have moved up or evaporated, and that results in a chain reaction with all water molecules moving upward due to their cohesion with the water molecules directly above them and their adhesion to the xylem walls. We can also look at root pressure. When soil moisture is high, root pressure will help move water upward a bit. And once water is through the epidermal layer of the root cells, you generally have enough pressure created to continue pushing the water through the endodermis and into the xylem at the center of the root. The cohesion tension theory, as well as the transpiration pull mechanism, has become pretty much the widely accepted explanation for water transport in plants. So not only do we have this kind of negative pressure that pulls water upwards, we also have a cohesion tension production because of root pressure that exists with the water pushing on the roots and absorption being promoted. And then we can also look at movement of substances in the phloem. We want to define translocation, which is a process by which organic substances move through the phloem of a plant. Things like food, glucose, They're all produced primarily in the leaves of a plant, so the food must move down from the leaves to other parts of the plant, including the roots. The roots are open repositories for excess food, they're good storage locations, so products of photosynthesis and other chemicals must be transported down the phloem in the plant when photosynthesis is occurring. So when photosynthesis is not occurring, the stored food must move out of storage and into the rest of the plant via the phloem. The currently accepted hypothesis as to how translocation occurs is the pressure flow hypothesis. That phloem cells must be alive because they actively expend energy to guide the flow of organic molecules through a plant. And the xylem cells, they can be dead because xylem essentially acts just like a tube only, and they don't need to promote the transport of water. So essentially, remember that xylem and phloem are the vascular tissue, and they can move water and minerals through the plant. Phloem is going to focus on photosynthesis products, things like carbohydrates, like the glucose made from the process of photosynthesis, and it must be moved up to flowers, buds, fruits, stems, leaves, but as well as down towards the roots. And then the xylem is going to move water primarily, but also stuff dissolved in that water. And it's mainly going to be just a tubular system that moves water from the roots upwards to the rest of the stem, shoots, and leaves, and even flowers. So we don't have the regulation in the xylem that we see for the phloem. That's why xylem can be or consist of dead cells, whereas the phloem can consist of living cells. So make sure you go back and review processes that ensure movement throughout the plant of certain substances like water. So make sure you go back and look at transpiration and some of the pressure hypotheses and know about the vascular bundles, differences between monocot and dicot venation. So just some things to go back and review. And next time we will look at part C.